In this section, we will quickly explore the term biotechnology. Biotechnology, as the name tells you, is using techniques or technology in for living organisms. And when we talk about biotechnology, you think of all of a sudden stem cells, major genetically modified foods, various crops, many hybrids, plants, and so forth. So all of that are examples of biotechnology. Biotechnology generally is implying a combination or recombination of DNA. So it is the tailoring of adding the desired characteristic on a genetic sequence, and when you do that, you pretty much have a perfection of the kind of organism you want. This is a nutshell what biotechnology is. Now the techniques that are involved in biotechnology are very detailed and very costly, and it is not a short time process. It takes days and days to get the series of experiment completed. The first part of introducing a DNA to a gene is to first chop the original sequence and then being able to insert your desired sequence. Chopping is done by these scissors, as they're called, and they are the restriction enzymes. They cut DNA into pieces. And another very important enzyme is a ligase, which then attaches the desired sequences with the parental DNA combination. So the ligase, you can consider that as the glue. So we have scissors and glue, and by which we can introduce regions of desired DNA. Now the term plasmid are usually a circular DNA, which are mainly derived from bacteria, and they have a very unique property that it can very easily and very quickly photocopy when it's introduced in a cell. So what happens is you take a plasmid, for example, this rounded DNA circular, and it is cut by certain enzymes. So when I make this cut, what will happen to this? It will become like a linear structure, and now I can attach my piece of my desired DNA and then use some sort of glue which can then glue these pieces together. So the glue that is used is the DNA ligase, and eventually you will have a sequence that is going to have your little inserted piece over here. So this is in a nutshell what biotechnology would do. There is examples of the same steps over here, but with better diagrams. You can see the scissors that are used are your restriction enzymes. I'm going to change the color over here, and you can see that the scissors are also cutting these plasmids, and then the plasmids and the DNA fragments are put together in this container, and they are mixed, in which then you will attach these are the lighter blue pieces. I hope you can differentiate with the darker blue, and you can see the light blues are in here introduced. So these segments are then introduced into a host cell, and it's usually bacteria, whereas the host cell divides, it automatically makes clones or exact copies of these foreign DNA. To make foreign DNA, a lot of times it is necessary to understand a little bit of biotechnology, and in this particular example I'm going to show you how HIV works. HIV is usually present in viruses, and it has RNA. It's usually an RNA virus, and when it infects a host cell, it undergoes a very unique process which is called as reverse transcriptase. Now remember, in the previous lecture we talked about DNA makes an RNA. In this case it's the opposite. An RNA makes a DNA, and this DNA then gets incorporated into the host DNA, and then directs proteins from this DNA. So now what has happened is this foreign object has introduced itself into the host DNA. And in this way, it disrupts the host cell's immunity, and the host cell's immunity just gets weakened during this process. Another term that you will hear about in biotechnology is the term of DNA sequencing. DNA sequencing is pretty much taking a section of DNA and sending it through an analyzer where it reads the different sequences of the nucleotides. And this will give some information that might be useful to compare, for example, in controlling diseases, to control and to understand what malfunction is there from the original disease. So what does a healthy cell look like, and what is a disease cell's DNA carrying for that particular disease? Another important tool that is used for biotechnology is called as fingerprinting. Fingerprinting is when you take a long strand of DNA and cut into shorter segments, and then each segment is separated by electrophoresis. Electrophoresis is similar to the concept of a sieve, and how a sieve will separate out the pastas. That's how the gel will separate out segments of DNA, and each DNA fragment is then going to be analyzed. For example, if there is a crime scene investigation, then in a crime scene, if there's certain evidence present that is shown over here, then this can be matched by the different sources of the reference that is used. So, for example, you can see over here, these are matching this one, so you can match the suspect to the evidence that is collected through the DNA. Last but almost there, genomics is a very broad branch. Genomics is a term that is used which implies the study of human genome. Human genome is a very large project, and human genomes showed that human genome has been sequenced, and there are 3 billion nucleotides in a human DNA sequence. Out of the 3%, 2% makes sense. The other 98% is just there for other reasons, also to depict variation during DNA fingerprinting. So an entire study is being done in which genomics is used to compare DNA sequences from different organisms, and this helps in developing new drugs, and pharmaceutical industry is really relying on comparative genomics. Lastly, we can see some of the products that are used that are formed by genetically engineered bacteria. Is genetically in medicine, agriculture, industry, and ecology. Usually, bacteria is the model organism used to study any of these diseases. Corn hybrid is produced that will give better yield through genetic engineering. Fish and mouse, for example, the green fluorescent that you see is present. I've actually bought fish from Pets West in the local Sierra Vista store that has the green fluorescent in them. And the pig and the monkey experiments that you can see over here also indicates genetically engineered organisms. So this is just a little bit of vocabulary word that I introduced you quickly towards these terms.